All right, all what I want you to have in your hand right now, I want you to have a piece of paper and a pencil, because it's really very important to understand these two lectures that we are going to talk about. The first one will be the cellular respiration, or how our cells harvest the energy. You remember the currency for the cell to do the job is ATP. From where this ATP came from? So this is what we are going to study right now. So I want you to have a piece of paper and a pencil, and then how the plants, who are the producer of the planet, produce those organic molecules that we are eating as animals to produce energy for the cell to do the job. So I want you to have a piece of paper. There are two important equations that we are going to study very deeply. The first one is the cellular respiration. The general equation for cellular respiration, which is an aerobic cellular respiration. That means it's needed to consume oxygen. We are going to make it very simple. This molecule or organic molecule is the organic fuel. We're making it as simple as C6H12O6. This is our organic fuel. This is what we are eating to get energy. We eat food. So let's simplify this food on a molecule of glucose, C6H12O6. We breathe oxygen, all right? And we produce CO2 plus water. This is what our cells do. So let's balance it. This is a chemical reactions that are the reactant that are the product for the cellular respiration. All right? This carbon came, of course, from this. This is a big molecules, all right? From big molecule, we come up to a small molecule. This is a catabolic reactions. This is where the sugar that is a big molecule, or this organic fuel, this food that you are eating, is going to be catalyzed into CO2, right? So this is a catabolic reactions. During the catabolic reactions, we produce energy. And this energy, the cells, the currency that the energy use from this organic molecule, it's the adenosine triphosphate. That's the currency that the cell use. So this is exactly what we are going to see in these chapters. We are going to study cellular respiration, which is an aerobic respiration, which is a catabolism reaction. And again, to simplify these reactions, we use the organic fuel as simplified as a molecule of a sugar, C6H12O6. So let's get started. So how do cells make ATP? They're in the process that we call cellular respiration. You know that animals, humans, we are heterotroph organism. We are the consumer of this planet. Who are the producer of this planet? The producer of the planet are the plants. This is because they do a process that we call photosynthesis that happen in the chloroplast using sunlight. So the source of energy came from the sunlight. And they produce an organic molecule. They produce the organic fuel that we eat as animals to produce ATP that will allow the cell to do the job. And this cellular respiration happens in an organelle in the cells that we call mitochondria. Mitochondria. So this is the global reaction for the cellular respiration. We go between parentheses. It's an aerobic respiration because it uses oxygen as a reactant. So this is an aerobic respiration. So I simplify this organic fuel as a molecule of glucose, since the glucose is the source of energy, right? Carbohydrate. So we simplified it, right? So we are going to use this. This is the general equation. The reactant are C6H12O6 plus oxygen producing CO2 and water and produce energy. This is a chemical energy. It's under the form of ATP for the cell to do the job, right? Here in this equation, we are going to see a molecule that is going to be catalyzed. This molecule, you see this carbon over here, the only source for this carbon came from this big molecule of glucose. So this has been catalyzed. So the C6H12O6 has been oxidized to CO2. Oxidized to CO2. That means it's going to lose electron. Reduction, we say reduction, which is diminution of the number of protons. Reduction, protons are positively charged, so therefore they gain electron. Reduction, so the oxygen will become a big molecule. You see, he gained electron. He gained electron to become a molecule of water. So I repeat it. C6H12O6 is going to be oxidized to CO2, and the oxygen, it will be reduced to H2O. When a big molecule becomes small, is oxidized because it's going to lose electron. He loses some electron. And oxygen, he's going to gain electron to become a molecule of oxygen. So the electrons are going to go from high energy and going down to a low energy. You know, during this process, when I have C6H12O6 becoming CO2, is going to reduce, is going to lose electrons, right? These electrons are not immediately going from C6H12O6 and then making CO2. It's a process, step by step. First of all, those electrons, this is very strong energy, electronic energy over there, guys. Those electrons are going to be captured by a system that they can resist this electronic energy. Everything is very smooth. The catalyze of the glucose into C6 into CO2, it's a step by step. And during this process, those electrons coming from here, they are not immediately going here. No, this is electronic energy. It goes step by step. Step by step. And they have some acceptor of electrons that we call them that they accept this high electronic energy coming from electrons. Those acceptors of electrons, they are carrier. We call them electron carrier. We have two that we are going to study here in this chapter. They have the nicotinamide adenine diphosphate, right? NADP plus. That once he accepts the electrons, and anytime when you say electrons, it's protons going down with it, because you have to have as minus as much as a plus. do not create any potential action. So when you have NADP plus to be reduced to NADH, so he accepts the electrons. Of course, the electrons are coming. The H plus also is coming. So they are going to be NADH. It's going to be reduced on NADH. So is the FAD plus too, which is also another acceptor of electron. The flavin adenine dinucleotide is going to accept also from FAD plus to become FADH2. So those are FAD plus and NADP plus are just carrier. They are going to carry this electronic energy coming from C6H12 before they're in the catalyze. He not only is going to be catalyzed, but every time you remember the first law of thermodynamics, the energy cannot be destroyed. Okay, any energy, we are not going to be losing it. Are not such a thing as energy lost. So those electronic energy that they are coming, those electrons coming from this big molecule of glucose are going to be carried by electrons carrier molecule. There are two, NADP plus that become NADH, and FAD that accept 2H plus to become FADH2. Right? I don't want you to retain the structure, but just to understand how it's works. Okay? So for the cellular respiration, I told you that we have aerobic respiration that require oxygen. And since the oxygen is present, anytime the oxygen is present, it's a redox reactions. That means one reaction, one molecule will be redox and the other molecule will be reduced. Anaerobic respiration does not require oxygen, and fermentation is anaerobic respiration. All this aerobic or anaerobic fermentations, they are exergonic reactions. This is catabolic reaction. They are going to produce energy. Does not require energy to happen. They are not endoorganic. Remember chapter 18, chapter 6, when we study the metabolism. Okay, so now let's go over those stages. You see those general equations that we have. This, it doesn't happen. It happened by steps. So let's look at those steps. The first one is the step number 1, which is the glycolysis. Glycolysis takes place in the cytosol or cytoplasm. Then we have an intermediate stage that's which will in an intermediate stage that will form acetyl coenzyme A. As soon as this, whatever came up from the glycolysis that take place in the cytosol will give me 2 molecules of 3 carbon pyruvate. If the oxygen is present, those pyruvate will enter the mitochondria. As soon as they enter the mitochondria, it's going again to be catalyzed in an acetyl coenzyme A. This is an intermediate stage. And after that, it's the acetyl coenzyme A that will enter the citric acid cycle. And here I am in the mitochondria. I'm not in the cytoplasm anymore. And then after that, they are also within this process over here. Remember, any times I am doing catalyzing, I am going to cut. I cut the glucose into 2 molecules of carbons. Those 2 molecules of carbons are going to be cut in an acetyl coenzyme A molecule. And after that, this acetyl coenzyme A will enter the citric acid to become a CO2. So I am going to reduce it to 1 carbon molecule of from organic molecule. I'm going to a gas molecule with 1 carbon molecule of CO2. So during this process, I am all the time releasing electronic energy, electrons. Those electrons are not going to circulate like that there. They are going to be captured, you remember, all the time by those molecules, by NAD plus and FID plus. They are going to be captured. And once they are captured all the time, either in the cytoplasm or either in the mitochondria, they are going to be by those molecules, they become reduced into NADH and FIDH2. Well, those molecules, they are going to give those electrons to what we call electron transporter chain. So this is grosso modo how cellular respiration happens. So we have, first of all, glucose. Remember what is the equation of this is why I told you to have a piece of paper. C6H12O6 plus 6O2 giving me CO2 plus H2O. So remember, I am catalyzing this molecule. This happens in the cytoplasm. This is the cytoplasm. This is the cytoplasm of the molecule. That's the mitochondria. Okay? This around here, these are cytoplasm. Okay? And this over here, that's the mitochondria. So first of all, the first stage, it's a glycolysis, right? Glycolysis, glucose, different step is going to give me pyruvate. Pyruvate is a 3-carbon molecule. I am going from a big molecule of glucose that is a 6-carbon molecule, it's C6H12O6, to a 3-carbon molecule. So that means I make from 1 molecule of glucose, I have to make 2 pyruvates, right? 2 multiplied by 3, 6. And then after that, I make the pyruvate. Pyruvate will enter by an active transporter into the mitochondria. Once entering the mitochondria, it will be oxidized into acetyl-CoA, and it's the acetyl-CoA which is an intermediate stage. It happens only if the oxygen is present, because glycolysis happens in aerobic and anaerobic respiration, both with the oxygen and without the oxygen. But if the oxygen is present, it will enter the mitochondria, and once he enters the mitochondria, it got immediately oxidized into acetyl coenzyme A that will enter the citric or Krebs cycle, right? Before I go any further, I want you to understand the 2 types of phosphorylations, because what we are having over here is to produce ATP, but these productions of ATP happen by two mechanisms, either by oxidative phosphorylation or either by what we call the substrate level phosphorylation. Okay, oxidative phosphorylations is the one that it's the one that produces almost 90% of the ATP generated by cellular respiration. It's happened using a pump, proton pump, ATP synthase pump. This is a proton pump, all right? This is oxidative phosphorylation, and it happens when the oxygen is present. The other type that produce also ATP, remember ATP is produced from ATP plus inorganic phosphate. It happens either using sensitized, which is an enzyme that is very sensitive. It gets stimulated by the passage of the proton, all right? And this is an enzyme, this is the ATP sensitized, or either by substrate level of phosphorylation. What's happened, this is another enzyme, but this enzyme, it recognizes substrate that is phosphorylated, and you will take the phosphate. Look at that. It doesn't need a proton. It doesn't need ATP synthetize. pump the substrate level of phosphorylation. He will take the phosphate and he will give it to ADP to produce ATP. This is what we call substrate level of phosphorylation. That means the phosphate to phosphorylate ADP came from another substrate. Phosphorylated substrate that we recognize have an active site in this enzyme. He will take his phosphate, produce product that is not phosphorylated, and he will give it to ATP to produce ATP. But this doesn't happen as often. This is the one that happens as often because it produces almost 90% of the ATP. So let's talk about the first stage now that we understand all this. Remember, to produce ATP, I need phosphate, ADP, I need ADP plus phosphate, either by oxidative phosphorylation or either by substrate level of phosphorylation. So we talk about the first stage, which is glycolysis. This is what we call between parentheses sugar cathine or splitting. Sugar cathine or splitting. I am going to catalyze. Remember, I have the word lysis, glycolysis. It means cause lysine, cathine. I am going to cut glucose. So I have my molecule of glucose, C6H12O6, and I am going to catalyze it. It happened only in the cytosol or cytoplasm. Does not require oxygen to happen. And it's divided in two phases. We have a phase that is investment phase, and we have a phase that is capture phase. In another word, it will consume some ATP to happen, but it will produce more ATP. It consumes almost 2 ATPs, but produce 4 ATP. So in the end, I will talk about the net production of ATP, which is 2. 4 minus 2 is 2. So let's go step by step about this. This is my molecule of glucose. All right, guys? I have an enzyme. You don't have to retain all this, but I want you to understand how it's works. All right? I have an enzyme. Look at that. He's going to phosphorylate this glucose. He's going to phosphorylate this glucose. So he consumes some ATP. Here is the first ATP he consumes. Right? And then after that, I have another enzyme that will convert it into fructose 6-phosphate. It's still 6-carbon molecule. But instead to be glucose 6-phosphate, he becomes fructose 6-phosphate. Look at that. Fructose 6-phosphate. He will be D-phosphate. Now I have 2 phosphate group. I am going to consume another ATP because ATP will be hydrolyzed into ADP, releasing inorganic phosphate, and this inorganic phosphate under the phosphofructokinase, this enzyme, number 3, it will phosphorylate this fructose. All right, then I came to the step 6. From this, I am going to catalyze it into B-phosphate glycerate. Look at that. 1, 2, 3. I have only 3 carbons on here on the glucose. And I am having, you see this number 2, I am going to produce 2 of them. 2 of those molecules. As soon realize that, guys. Look at this. As soon as I am coming in a number of carbons that is less, I am releasing electronic energy. And this electronic energy is not going to see electrons circulating around. No. So I am producing 2 of this 1,2-B-phosphoglycerate. Right? It's a B-phosphate. It's a phosphate here and phosphate here. As soon as they came into 2 carbon molecules, I am releasing some electron energy. And who are the capture for electronic energy? The NAD+. I'm going to capture this electronic energy, those electrons. I am not going to let them circulate like that. So I'm going to capture them, store them. They will become reduced to 2 in ADH. So I am producing 2 in ADH. Let me change this color. I am producing 2 in ADH, right? And after that, I am going to take off one phosphate group. So I'm going to reduce on size. Anytime I reduce on size, I'm producing energy, right? So this phosphate will phosphorylate ADP. So I'm producing 2 ATPs. Since I have 2, it's 2 of this molecule. So 2 phosphates with 2 ADPs produce 2 ATPs. Again, I continue. So I have already from previously, I'm going to work 2 in ADH and 2 ATP from previous slide. And then I keep going until I produce pyruvate, have no phosphate. And again, because I have always 2 of phosphoenol pyruvate, so I am going to phosphorylate the 2 ATP to 2 ATPs. So far, I produce 2 in ADH. I'm not a good writer, am I? And 4 ATP. 4 ATP. Remember, this is what I have so far, but I did use 2 ATPs. I did from the beginning. Remember, I used 2 ATPs. I show you even the slide that was 2 ATPs. When I start phosphorylating my glucose, and then transform my glucose to fructose, I use 2 ATPs. So in the end, net, what I produce 2 pyruvate per molecule of glucose. I produce 4 minus 2, 4 ATPs minus 2 that I use form. It's 2 ATPs net, and they produce 2 in ADH. So summary for this step, which is the glycolysis. Glucose is catalyzed with different enzyme into 2 pyruvate molecule of 3 carbons, producing output is 2 ATPs and 2 in ADH. So the glycolysis reactant is glucose, right? And it happened with oxygen or no oxygen. This is always happen. And as a product, I produce 2 pyruvate, 2 ATP, net 2 ATP. But I do produce 4 ATPs. But if I said the word net, it's only 2 ATPs. So don't confuse it. The word net is very important to be used. And 2 in H. We finished the first step. That's my step 1. Okay. My step 2 is the intermediate. What's going to happen? So let's see. Because the 2 ATPs is one of the, this is what I want. Is I want my cells to harvest this energy that it came from this organic molecule of food that I am consuming. So this is good. I have 2 ATPs per molecule of glucose. I still have those electronic energy that my cell have to harvest too. I have to use it because my cell does not recognize this as currency for her to do the work. It recognizes ATP as currency. It doesn't recognize those carrier electronic. It's very strong in electronic energy, but my cell are not going to recognize in ADH as currency for them to do it. The currency that you know how to use or what to use is only ATP. So far, I produced 2 ATPs. How this ATP is produced, guys? All right, let's see. Let's see how this ATP is produced. This ATP is produced by this substrate. Using this enzyme, it's by substrate level of phosphorylation. So is this one too. So far, so this ATP, in conclusion, is produced by substrate level of phosphorylation. So glycolysis produces 4 ATPs by substrate level for phosphorylation. Anytime he take only the phosphate from a phosphorylate substrate, you get it? Just look at your steps. All the time, this is what they are doing. They are just taking phosphate from a substrate. All the time. This is what they are doing. There are no oxidative phosphorylation whatsoever. That means the pump, the ATP synthetase, the enzyme is not used. Did you see the word ATP synthetase anywhere? No, it's not being used. It's only substrate level phosphorylation. I take this phosphate from this substrate to produce this ATP all the time. So now the second step. The second step is if the oxygen is present, happen. If the oxygen is happen, what's going to happen to this pyruvate that has been produced in the cytosol? You will enter, look at that. It's an active transporter, pacylated transporter, because it's big to enter. The double, remember that the mitochondria, it's an envelope. You have double membrane. You have an inner membrane that is in contact of the matrix. That's the matrix for the mitochondria, right? And this is the outer membrane that is in contact of the cytosol. Okay? And between them, it's the intermembrane space, this dark orange over here. That's the intermembrane space of the mitochondria. Okay? So we have the inner membrane. That's the inner membrane that is in contact of the matrix. That's the matrix. Right? And this is the outer membrane that is in contact of the cytosol of the cells. Right? You have to know this. Okay? The intermediate stage happens when the oxygen is present. As I said, the pyruvate will enter the mitochondria. As soon as he's inside of the mitochondria, what's going to happen is going to be oxidized again to form an acetyl coenzyme A. And when he forms an acetyl coenzyme, look at that, acetyl coenzyme A, it's very unstable molecule. You have only 2 carbons. And it's got stabilized by this coenzyme, the acetyl CoA, right? This is very unstable. It will enter, we will see that immediately will enter the citric acid. And during this process, what's going to happen? Because I have 3 carbons molecules, giving me 2 carbon molecules, he will produce a CO2. And also because he's going from big molecule to a small molecule, I told you it's always some electronic energy circulating, there will be captured by NADH. Remember, how many pyruvate I have per molecule of glucose? I have 2 pyruvate, but those is 2. So 2 of them are going to enter to produce 2 CO2 and 2 acetyl coenzyme A and 2 NADH. No ATPs is produced here. Yeah, one thing is very important. You remember our general equation for cellular respiration. What is it? Post it again. This doesn't exist anymore. It's gone. It's been catalyzed. Now it's carried by the pyruvate. The energy that was here, now it's within the pyruvate and within also the NADH. And some of them, it's already producing ATP. Right? But once he entered the acetyl, the pyruvate entered the mitochondria is oxidized into acetyl coenzyme A. And this acetyl coenzyme A, in this process, it produces CO2. It produces 2 CO2s. And CO2, what is it? It's just one of the products. You know, sometimes you are going to see your aunt that is in Fort Worth, and you have a friend that live in Arlington, and you are coming from Dallas to Fort Worth, and you tell your friend, oh, I'm going to see my aunt that is in Fort Worth. and your friend will tell you, oh, stop by, because your friend is in your way to Fort Worth, because Arlington is between Fort Worth and Dallas. So your friend will tell you, oh, stop by BY. So this, did I finish my equation? Did I have water formed? Not yet. So the CO2 therefore has been formed as what we call the byproduct. Byproduct is a product that has been formed before even my equations or my reactions finish. It's a byproduct. BY, byproduct. What's going to happen to the acetyl coenzyme A? Now the acetyl coenzyme A that has been formed is ready to enter the citric acid or the Krebs cycle, and is going to be the feed, is going to be the source that is going to complete my oxidation of the glucose that I start from the cytoplasm. Right? So let's look at the Krebs. So I have my acetyl coenzyme A. Remember that I formed two because I have two pyruvate. You will look at that. It will be formed. It will combine with an oxaloacetate forming a citrate. And the next step are going to be just to decompose the citrate back. This is a 6-carbon molecule, but it's very unstable. It is. It will combine with the oxaloacetate to form a citrate, which is a 6-carbon, but it's very unstable. All of it, I don't want you to retain all this. All of it is just going to have the citrate, decompose the citrate back to oxaloacetate, so he can combine with another acetyl coenzyme. The most important here is what we produce from the cell cycle. All the time I am producing NADH, but they have also some like 16-8 to fumarate. They are not going to give their electrons to NADP plus to form NAD. They're going to give it to another. They have affinity to give it to another acceptor of electrons, which is the FAD plus. So during the cell cycle, the citric acid cycle, per molecule of pyruvate, I form 3 CO2, 3 NADH, 1 ATP, and 1 FADH. So per molecule of glucose or per molecule of 2 pyruvate, I produce 6. You have to multiply everything by 2 now. 6 CO2, 6 NADH, 2 ATPs, and 2 FADH. These 2 ATPs, it's only by subtract level of phosphorylation. If you don't trust me, look at it. It's only, so let's look at where the ATPs is being formed. This is just a phosphate that has been taken by phosphate level of phosphorylation. There are no oxidative phosphorylation. So this is what I got if I combine everything per molecule of glucose from the beginning. I have 2 pyruvate. The 2 pyruvate enter. So in the beginning, I have 2 ATPs, 2 NADH. Then after that, I have 6 CO2, 6 NADH, 2 ATP, and 2 FADH. My problems now. Did I have H2O? Not yet. It's one of the final products of my chemical reaction, C6H12 plus O6, giving me C6O2. C6CO2, it's finished. That's it. I finished catalyzing my glucose. It's been oxidized to C6CO2. But I still have energy. I still doesn't form my water. And I still have this electronic energy, this FADH and NADH. Those are going to give their electrons only to a molecule that they are very sensible to accept. They are very strong enough and very sensible to accept those electronic energy where those molecules exist. Those molecules, this is what we call transporter electronic chain. They exist in the inner membrane of the mitochondria. Remember, the mitochondria is the inner membrane. That's the yellow thing that we saw, and the blue thing is the matrix, and this part, the red part, is the outer membrane. And here is your cytoplasm of the cell, for example. Okay, so the inner membrane have those electronic transporter chain. They are just molecule. I don't want you to name them. All right. They are transporter 1, 2, 3, 4, and between 3 and 4 we have cytochrome C. So those they are proteins. If you see, they are protein that's on the membrane, and this is my inner membrane of the mitochondria, and this is my outer membrane of the mitochondria, and between them I have my intermembrane space. All right, pay attention, please. Those electron transporter electrons, they are only going to realize following this electronegativity. I have complex 1 is less electronegative than 2, 2 is less electronegative than 3, 3 is less electronegative than the cytochrome C, cytochrome C is less electronegative complex of 4. And the oxygen, because I am going to reduce oxygen into water, have affinity to accept electrons only from this, from this complex 4. You have a sensibility. The oxygen will not accept electron from here, not from here, not from him, only from the complex of 4. It's just the way it is, guys. So we will accept electrons coming from here. So let's look at how these electrons are traveling. All right, let's look at it, how they are traveling. Stay with me. It's very beautiful. These chapters, I'm crazy about it. Very crazy about it. Look at it. I have the NADH. I have sensibility to give the electrons to the less electronegative first, which is the complex 1. So he will give him the electrons. Okay, you can take the electrons from me. I will become NADP plus again. This is how it circulates the NADP plus. All right, I become NADP plus. But this guy is more electronegative than this one, so we will attract those electrons coming from here. All right. And this is more electronegative. He will give it to them until he will give it to the final acceptor of electrons, which is the oxygen. But there are one thing that happen, guys. Remember what I told you. When an electron circulates to not cause any potential action, I will to not change the voltage of my membrane. I always have as much electron circulating, as much proton circulating. Positive, negative, neutralize each other, right? So anytime my electrons is going, I will put it in blue or blue is going. That's my electrons until he end up in the final acceptor of electrons, which is the oxygen. In the same time, what's happened? Proton is circulating. Proton, look at that, is going out. He's not going to go to complex 3. Only electrons are going to go to complex 3. Only electrons are going to complex 4. All right? So he's going to go out, proton. Oh my God, I found the problem, guys. This part of my membrane, this side that is in contact of my membrane, remember, it's a bilayer. So this is the matrix of my mitochondria over here. And this is my intermembrane space. And I am at the inner membrane of the mitochondria. It's going to be more positive. I will create an electrochemic gradient of concentrations over here. I don't want that to happen. I don't. I'm in problem now. Now I have more positive charge in this side of the membrane. They need to come back and vanish inside of the matrix. Those protons need to cross this membrane. But remember, my membrane, the problem is, it's a very selective membrane. Not even a proton can cross it easily. They need a transporter. They are charged positive. They are going to deal with this fatty acid sandwich over here. So they need a transporter. The only transporter for proton is what? ATP synthetize. ATP synthetize this guy over here, guys. ATP synthetize. ATP, it's an enzyme. The rotor, which is this guy, the rotor, which is this, is in contact of the matrix. And ATP synthesize her job anytime it gets stimulated, it will phosphorylate ADP to ATP. So anytime protons going in, stimulate the ATP, ADP will be phosphorylated to ATP. Proton going through, ADP will be, this is what we call oxidative phosphorylation. Oxidative phosphorylation. I have some video over here that I want you to, and I really want you to review this. Review it very, very nicely. Forcible glycolysis that happen in the cytoplasm, pyruvate enter the mitochondria by facilitated active transporter. Once in the mitochondria, it gets oxidized to acetyl coenzyme A. Acetyl coenzyme A is the one that will feed the cell cycle. It will combine with an oxaloacetate to form a citrate, which is a 6-carbon molecule that is very unstable. And during this process is going to be reduced, is not only is going to produce the oxaloacetate, but reduce into CO2. So my oxidation of the glucose is finished because I form my C6 CO2, but I still have, and I form ATP by phosphorylation, by substrate level of phosphorylation, the electronic energy now, the NADH2 are going to give those electrons to those transporter that exist in the inner membrane, of the mitochondria. That's the inner membrane, that's the outer membrane, and that's the intermembrane space. And by doing that, right, they are going to chase the proton outside. So this part of the membrane become more positive than this part. So they have to come back. And the only way to come back is by ATP synthase. And anytime proton cross to establish the potential action of this membrane, the proton have to cross this pump to be vanished, to be dissolved into the matrix. This stimulates this the ATP synthase, and therefore they will produce more ATP by oxidative phosphorylation. I mean, other source of energy, not only I simplified it as a molecule of glucose, but they can be proteins, they can be lipids, they are all, it's almost the same fat, they are almost the same process, right? For a carbohydrate, it's glycolysis. For proteins also, we are going to reduce it to amino acid. It's the same process. We're not only eating carbohydrate, right? Now, let's look at anaerobic respiration, and I focus on two anaerobic respiration. I focus on the two types. One is alcohol fermentation and lactic acid fermentation. If you look at, I done one of anaerobic alcohol fermentation in one of my video with using yeast and sugar and using balloon. And when the balloon are blowing up, that's mean I am producing CO2. And this is one of the products, and I like to show it as a picture. So this is the alcohol fermentation and lactic acid fermentation. Remember, glucose gets catalyzed or oxidize with or without the oxygen. It's happened all the time. Glycolysis for the alcohol, it give me 2 pyruvate, right? And they produce 2 ATPs. I produce 2 NADH, but it's circulating. In the end, I produce alcohol, 2 ethanol, right? And you can smell it when you do this yeast with the sugar. You can smell that it smells bready, it smells alcoholic smell. For the lactic acid, I produce 2 pyruvate, true, I produce 2 ATP and 2 lactate, but I don't produce any CO2. CO2 is produced only during the alcohol fermentation. And how I know that the CO2 is produced? Because I saw my balloon blowing up. If you look at my fermentation, alcoholic fermentations video in my YouTube channel. So as a practice, you have this quiz to practice, and that's it. And I will see you tomorrow for the next chapter about the photosynthesis now. Stay safe and healthy, and we'll talk later.